Hello everyone, my name is Dr. Sikandar and today we will discuss about the structure and the function of lungs, like how blood gas used to go, and this is based on the physiology. So yeah, chapter 1 of physiology in the lungs. So let's start. What about the blood gas interface? So yeah, transport of like blood gas is usually like it is done by simple diffusion, that is from an area of high to low partial pressure. So here in lungs, like it is done by simple diffusion from high pressure to low partial pressure. So a fixed law of diffusion states that amount of gas that moves across a sheet of tissue is proportional to the area of sheet but inversely proportional to its thickness. So it is inversely proportional to the thickness of the sheet and the directly proportional to the area of that sheet. So yeah, if area of sheet will be more, then diffusion will be more. If thickness will be less, then diffusion will be more. So it's like that. Blood gas barrier is exceedingly thin and has an area of around 50 to 100 square meter. That is like huge area. Therefore, well suited for gas exchange. So in like our lungs, blood gas barrier is very thin, like it is exceedingly thin and has an area of 50 to 100 square meters. Therefore, it is well suited for gas exchange. So, area is created by wrapping the small blood vessels, like capillaries, around enormous number of small air sacs, which are called alveoli. So, there are a lot of capillaries around the alveoli. That's why this area is created for diffusion. There are around 500 million alveoli in human lungs. So, 500 million alveoli in human lungs are there, and each about 1 by third, like it is 1 by 3 mm in diameter. If they were sphere in shape, their total area will be around 85 square meters, but their volume is only 4 liters. So the volume of lungs is only 4 liters, and like total surface area is around 85 square meters. If we think like it is 500 million alveoli in human lungs are there, and like each alveoli is having 1 by 3 mm of diameter. By contrast, a single sphere of this volume, they are telling like alveoli has been divided in 500 million alveoli. So if it will be single sphere of same volume, then it will create only 1 by 100 square meters. That is very, very less. Thus, lung generate this large diffusion area by dividing into a myriad of units. So, because it used to divide in 500 million alveoli, that's why it used to create that much of area, like 85 square meters of area. So they are telling the alveoli are not spherical in shape, like in practically they are polyhedral in shape, and they are telling like nor the whole of their surface is available for diffusion. So like the whole surface of alveoli is not available for diffusion. So the partial pressure of a gas is found by multiplying its concentration by the total pressure. Obviously, like what is the concentration of alveoli in that chamber and what is the total pressure of that chamber. Then we can easily get the partial pressure of that gas. For example, dry air is having 20.93% of oxygen, and its partial pressure is at the level of C will be like 20.9 divided by 100 into 760. 760 is the barometric pressure, surrounding pressure. So it will be around 159 mmHg. They are telling when air is inhaled into the upper airway, it is warmed and moistened, and the water vapor pressure is around 47. So the total dry gas pressure is only 760 minus 47. We have to like minus that pressure also. So like actual like jo dry pressure will be around 713. So according to 713, like the partial pressure of oxygen will be 149. So we have to divide minus this 47, because like yeah, our airway is having this much of water vapor pressure, 47 mmHg. Then they are talking about the electron microscopic appearance, which is showing a pulmonary capillary in the alveolar wall. So we can see like this is an alveolar wall, and here we can see this whole thing is like a capillary. This is the epithelium of the lung. Here we can see this is the capillary, which is having like this capillary is having RBC and the endothelial cells. And yeah, we can see like how which things comes in between. Like so, first thing is like if oxygen is traveling to RBC, then first he will go through epithelium of the alveoli, then basement membrane of alveoli, then it will go into the interstitium. So this part is interstitium. Then basement membrane of endothelium of this capillary. Then it will go to endothelium. Then it will go to plasma. Then finally it will go to RBC, inside the RBC. So these things come in the like blood gas barrier. So yeah, airway and airflow. Airways is a series of branching tubes which become narrower, shorter, and more numerous as they penetrate deeper into the lungs. So they are telling like As they used to divide, they used to become narrower, shorter, and more numerous as they penetrate deeper into the lungs. The trachea divide into right and left main bronchi, which in turn divide into lobar and finally into the segmental bronchi. We know like every lung is having 10 segments. So finally it used to divide into segmental bronchi. This process continues down to the terminal bronchi. So like they used to make like left and right main bronchi, then lobar, then segmental, and finally till the terminal bronchiole, which are the smallest airway without alveoli. All of these bronchi make up the conducting airway. So this is like this whole part from the trachea to the terminal bronchiole is the conducting airway. The larger proximal area have a lot of cartilage in their wall. As the airway progresses distally, the proportion of cartilage decreases and the smooth muscle cells increases, such that the very small distal airway are composed mostly of the smooth muscle cells. Very, very important. So they are telling like finally the like terminal bronchioles are made up of only smooth muscles. They do not have any cartilage because the conducting airway opens into they are telling like conducting airway do not have any alveoli, so there is no exchange there, and like it will contain anatomic dead space, where the term dead space refer to area of lung that receive ventilation but there is no blood flow there, and its volume is around 150 ml and it does not participate in the gas exchange. So this is the anatomic dead space. So here we can see a diagram which is showing this is like terminal bronchiole, and these are the alveoli, small alveoli are there, and like inside these alveoli we can see like small pores which are pore of Kohn. And here if we will remove the part of alveoli, then it will look like this. This is the picture of lung which looks like that. Like this is the part which is the conducting zone. So here we can see another diagram which is showing conducting zone and the transitional plus respiratory zone. So this is the conducting zone. Conducting zone is having like 1 to 16 divisions. So first thing is like 0 is trachea. Then comes first thing is bronchi. These bronchi used to divide. Then finally they used to form bronchioles, and finally they used to form terminal bronchiole. So terminal bronchiole is 16th number of division. After that comes respiratory bronchioles, like which makes 17, 18, and 19. Then comes alveolar duct, which makes 20, 21, and 22. And like finally, there comes alveolar sacs. So yeah, it is like that. And this is 20 number 23. So 1 to 16 are conducting zone, and after 17 to 23, these are transitional plus respiratory zone. And respiratory bronchioles make 17, 18, 19. Alveolar duct makes 20, 21, 22, and then alveolar sacs make 23. They are now they are talking about the like cross sectional area and like airway division. So till like we can see like airway generation 15 or 16, around like 16 and 17, there is conducting zone, and conducting zone is having like very small cross sectional area till here like cross sectional area is around like 100 or like around 120, 150. But from here it used to increase like exponentially after like 15th or 16th generation. It used to grow exponentially, and like from 16 or 17th generation we can see this used to grow like 200, 300, 400, 500 centimeters square. So diagram is showing the extremely rapid increase in the total cross sectional area of the airways in the respiratory zone. So in respiratory zone suddenly lung these cross-sectional area of this airway used to increase. As a result, the forward velocity of gas during inspiration become very small because the area is very large. So this like speed will become less. And region in the region of respiratory bronchiole, and gaseous diffusion becomes chief mode of the ventilation. That's why like here diffusion used to occur. Here we can see this is the view which is showing like how capillaries are there in our lungs. View of alveolar wall showing the dense network of capillaries, a small artery and a small vein. So yeah, they are telling like this is the small artery, this is the small vein. Can be seen the individual capillary segment are so short that the blood forms are almost continuous sheet. So like here capillaries are making like very small units, and finally they are opening in veins. But like this network is very like almost continuously making almost continuous sheet, very thin. So yeah, this is another diagram which is showing microscopic section of dog lung showing capillaries in the alveolar wall. So yeah, we know like this is the alveolar wall. Yeah, these are alveolar walls, and inside these alveolar walls blood gas barrier is so thin that it cannot be identified here. The terminal bronchiole will open into the respiratory bronchioles which have occasional alveoli budding from their walls. they will open in alveolar duct and completely lined with alveoli. So respiratory bronchiole will open into alveolar duct and finally they make alveoli. These alveolated regions do gas exchange and this is known as respiratory zone. The portion of lung distal to the terminal bronchiole forms an anatomical unit called acinus. So distal to terminal bronchiole, the part of lung is known as acinus. So this is the anatomical unit of lung. The distance from the terminal bronchiole to the most distal alveolus is only a few millimeters, but the respiratory zone makes up most of the lung. Its volume is being up to 2.3 to 3 liters, 2.5 to 3 liters during rest. So the volume of respiratory zone is 2.5 to 3 liters, while total lung volume was 4 liters, around 4 liters. Very, very important, around 4 liters. But the volume of respiratory zone is 2.5 to 3 liters during rest. During inspiration, the volume of thoracic cavity increases and air is drawn into the lungs. Increase in the volume is brought about partly by the contraction of diaphragm, already we know, which causes its descent, and partly by the action of intercostal muscles, which raises the ribs and increases the cross-sectional area of thorax. Very, very important. So they are telling like this is our lung. By diaphragm, it used to go like this, and by intercostal muscles, it used to move like this. So intercostal muscles increase the width of lung, and like this diaphragm used to increase the vertical diameter of lung. Inspired air flows down to about terminal bronchioles by bulk flow, like water through a hose. Beyond that point, the combined cross section of airways is so enormous, it is so like high because of the large number of branches that the forward velocity of gas become very small. So like yeah, we know after the respiratory bronchiole or after the terminal bronchiole, in the respiratory bronchiole, like velocity used to be very, very low because of like high large number of branches and high cross-sectional area. So diffusion of gas within the airways then takes over as main mechanism of ventilation in these respiratory in this respiratory zone. The rate of diffusion, so yeah, they are telling the rate of diffusion of gas molecule within the airway is so rapid and the distance is so short that the difference in concentration within acids are virtually abolished within a second. So it is the game of only seconds, fraction of seconds, because like it used to go, these used to diffuse very rapidly. However, because the velocity of gas falls rapidly in the region of terminal bronchiole, we have already discussed, inhaled dust particles frequently settled here. So they are telling like inhaled dust particles usually settles in the terminal bronchioles. Very, very important concept. The lung is elastic and returns passively to its pre-inspiratory volume during resting breathing. It remarkably easy to distend. A normal breath is about 500 ml. Obviously, tidal volume is around 500 ml. For example, require a distending pressure of less than 3 centimeter of water. By contrast, a child's balloon may need a pressure of 30 centimeter of water for the same change in the volume. So they are telling like it requires very small amount of the pressure to distend these like alveoli. The pressure required to move gas from the airway is very small. During normal inspiration, an airflow rate is 1 liter per second require a pressure drop along the airway of less than 2 centimeter of water. So yeah, it requires very less pressure, like only less than 2 centimeter of water. Then they are talking about the blood vessels and flow. So pulmonary artery used to open in the capillary. Then finally they used to take pulmonary vein used to take the blood away. Initially, the arteries, veins, and bronchi run close together, but toward the periphery of lung, the vein move away to pass between the lobules, whereas the arteries and bronchi travel together. As we know, like arteries and bronchi used to travel together down the center of the lobule. So they used to go to center of lobule and like vein used to go away from the lobule. The capillary form a dense network in the wall of alveoli. We have already discussed. Diameter of a capillary is around 7 to 10 micrometer, just large enough for a red blood cell. So red blood cell is having like almost this diameter, like it is just large like to red cell. The length of the segment are so short that the dense network form an almost continuous sheet of blood in the alveolar wall. A very efficient arrangement for the gas exchange. Yeah. The extreme thinness of the blood gas barrier means the capillary are easily damaged. So increase in the pressure in the capillaries to high level or inflate to the high level or inflating of lung to high volume will cause the stress in the wall of the capillary to the point at which ultrastructural changes can occur. And the capillary then leaks plasma and even red cell come into the alveolar spaces. Very, very important. Then they are telling the pulmonary artery receive the whole output of right heart. But the resistance of the pulmonary circuit is astonishingly very small. A mean pulmonary artery pressure is only 20 centimeter of water, about like 15 mmHg is required for flow of 6 liter per minute of blood. Each red cell spend about zero point 0.7 seconds in the capillary network. During this time, probably traverse 2 or 3 alveoli. So like they are telling like these red cells used to like traverse only 2 to 3 like these yeah 2 to 3 alveoli. The lung has additional blood system, the bronchial circulation that supply the conducting airway mainly. So like bronchial system used to supply conducting zone, and pulmonary system used to supply take like participate in this gas exchange. So yeah, they are telling like bronchial circulation that supply the conducting airway down to about the terminal bronchiole. Like they used to come till terminal bronchiole. Some of this blood is carried away from the lung by the pulmonary vein, and some enters in the systemic circulation. Yeah, the flow through the bronchial circulation is mere fraction of that through the pulmonary circulation, and the lung can function fairly well without that. Like they are telling, if bronchial system is not there, even then this lungs can work easily. So the example of this is following the lung transplantation, lungs function very well. Removal of inhaled particles. They're telling like lung has largest surface of body to an increasingly hostile environment. So it used to make a hostile environment. Various mechanisms for dealing with inhaled particle have been developed. So they are telling large particles are filtered out from the nose itself. And smaller particles that deposit in the conducting airway are removed by moving a staircase of mucus that continuously sweep debris up to the epiglottis, where it is swallowed. They are telling the mucus secreted by the mucus gland and also by the goblet cells in the bronchial wall is propelled by millions of tiny cilia, which move rhythmically under normal conditions, but they can get paralyzed by some inhaled toxins. So then they can create the disease. The alveoli have no cilia, but large wandering cell called as macrophage used to be there. The foreign material is then removed by the lung, by the lymphatics, or by blood flow. Blood cells such as leukocyte also participate in the defense reaction to foreign material. So this was all about the initial first chapter of physiology. That's it. Thank you guys.